and we can kick it off now that hopefully most people have logged in and can see us. Good evening to everyone who's joined. And I'm just going to give you a quick orientation about what the setup is in this webinar for people who aren't familiar. If you have a question during the meeting, you can raise your hand you can raise your hand. You can use the Q&A box, which is in the bottom right hand. So we'll try to answer questions during the meeting as much as we're able. And we're going to just follow the agenda, as I hope you've all seen on the Eventbrite link. So I'm going to start the meeting now. I'm going to share my screen. I hope you can see my screen. Okay, is everybody able to see my screen? Can one of the panelists just tell me there?
Yep, I can see it.
Okay, great. Okay, wonderful. So I'm just going to start now. Welcome everybody to this evening's event. We're going to talk about the results of the Darwin Plus project. Where are they now? Right whales in South Georgia waters. And this is a big initiative. It's been going on since 2016, and it received funding from Darwin Initiative, from the EU BEST, from South Georgia Heritage Trust, Friends of South Georgia Island, World Wildlife Fund UK, and logistical support from the government of South Georgia and the South Sandwich Islands. So thanks very much to all of those. Please do put any questions that you have about the talks into the Q&A box, and we'll try to answer them during the sessions. So this project, as I said, it began in 2016, and it was initially on right whales, and I'm going to give you a quick orientation about South Georgia first. Here's South Georgia. It's in subpolar waters in the high latitudes of the South Atlantic, and you can see the purple lines on this map, which are polar frontal zones, major oceanic fronts. And what you can see with South Georgia is it actually has, it sits next to the Antarctic circumpolar current, and the front actually surrounds South Georgia on 3 sides. Now, as a consequence of where it sits, it receives huge quantities of Antarctic krill. Krill are born in sea ice, and so they are recruited, they're born in sea ice, and then they come into subpolar waters on the Antarctic circumpolar current. And large quantities of krill get to South Georgia, and it's evidently, because of that, a really important place for lots of krill predators. Penguins, seals, and other seabirds, and of course whales. South Georgia was a really important place for whales. Many of you who've seen us talk before will have seen this slide, but it goes without saying the importance is really shown by the fact that over 176,000 whales were killed just within a day's sailing of South Georgia. It was such an important proportion of the entire Southern Hemisphere catch during modern whaling that it represents nearly 10% of all the catches made in the Southern Hemisphere. So with over 176,000 whales killed, we know that this was a really crucial place for whales to feed. Right whales in the Southwest Atlantic. Now, they were less hunted during the modern period of whaling because they were actually wiped out mostly by the 1850s. Right whales have been exploited in the South Atlantic for 350 years, starting in the 1600s. They were protected for much of the 20th century. Their numbers were so low that they weren't very heavily hunted. And now they're beginning to recover. So there's signs of population growth on calving grounds across the South Atlantic, in Brazil and Argentina and South Africa. We also know from work that's been done in Argentina and Brazil that calving rates for right whales have been linked to high latitude climate. And we think that high latitude climate is basically a proxy for the availability of krill, which they feed on at high latitudes. We also know that there's been large numbers of dead calves reported in Argentina since 2003. This the calving ground Peninsula Valdes in Argentina is a key calving area associated with the South Georgia feeding ground. So one of the 3 hypotheses as to why there are so many dead calves turning up during calving season is that perhaps variation in food availability might be influencing the health of the mothers. Another thing that really motivated us starting to focus on right whales at South Georgia is that until the mid 2010s, so 2015, they were the most commonly seen whale species in South Georgia waters. So our project aimed to establish baseline data on the status and recovery levels of right whales on South Georgia feeding grounds after whaling. We specifically had quite an ambitious target to characterize the abundance, distribution, diversity, calving ground connections, habitat use, exploitation, and health of right whales in order to help the government and other associated bodies studying whales in the South Atlantic do the best possible conservation management. This was a big project, and it involved a large number of institutes who have contributed their time and scientific expertise and their expeditioners to join our voyages. So I want to say a huge thanks to all of the institutes involved. In South Georgia, we maximised the data collection that we could make on every single voyage. As well as using observers to site whales on expeditions, we also used acoustics, and Susie Calderan is going to talk about that this evening in more detail. But the acoustics also helped us to detect whales at sea because we could listen to them calling and find out the direction that they were calling from. So whilst using observers and acoustic detectors, we could then work on individual whales. We collected blow samples in order to characterize the health of the animal via its microbiome. And you'll hear more about that tomorrow from Carolyn Miller. We also collected overhead images of the whales to study their body condition, and there'll be more about that tomorrow as well. We collected photo IDs of individual whales in order to compare the photographs from South Georgia with calving areas across the South Atlantic. And again, you'll hear more about that both today, mostly today. We also collected fecal samples where possible in order to understand diet. And we tracked 2 whales in South Georgia waters, and Amy Kennedy is going to talk about that today. We collected skin samples. To date, we've collected 10 skin samples, and we're using those to study the genetics, the foraging ecology by looking at the skin chemistry and isotopes, and also hormonal profiles of whales in South Georgia. And you're going to hear more about all of those studies tonight. Now, in terms of abundance, we were rather disappointed because we never saw enough whales, right whales, to get a good estimate of abundance. But I can give you a sense of what's happening for right whales over time in South Georgia waters. This graph on the left shows what the South Georgia Museum has been compiling since 1992, and that's the counts of whales sighted and reported to the museum between 1992 and 2018. What you can see here is that while humpback whale numbers have been increasing since 2013, the southern right whales in blue have stayed very, very steady over 25 years in South Georgia. So despite a rise in numbers on their calving grounds, the numbers of right whales using South Georgia doesn't really seem to have changed in terms of reports to the South Georgia Museum. We also compiled information from the fisheries patrol vessels. You can see the diamonds here, and again the blue diamonds show southern right whale sightings, and you can see that they really haven't changed much in over 15 years. And what did we see on our surveys in the last 3 years? Over 3 years of different surveys conducted at South Georgia, of which the Darwin surveys were the 2018 and 2020 surveys, you can see that over huge distances, there was some variation in the number of right whales seen per nautical mile. So in this case, for example, in 2018, we saw a lot of whales. We saw 31 whales over just over 300 nautical miles operating from a yacht. And that equated to about 0.09 whales per nautical mile then 2 years later, in 2020, the Braveheart survey went around the island twice, and they did nearly 3 times as much survey effort, and they saw only 13 whales using acoustics and sightings, suggesting that 2 years later, we had about 1/10 of the right whales using South Georgia waters. So we can see that there are real variations in the numbers of whales that choose to use South Georgia each year. So whilst this is a qualitative comparison, I think it's quite clear to say that whilst we know southern right whales do regularly visit South Georgia, and this map, for example, shows a lot of sightings that have been reported to the museum by the fishery patrol vessel and by our own sightings, we know that abundance is varying substantially between years, which points to the fact that feeding grounds in other parts of the Scotia Arc and Patagonian shelf are clearly important for this population. So despite mixed study results on abundance, we have been able to collect a lot of data on individual whales, and that's what the team are going to tell you about over the next couple of hours. I just wanted to say a huge thanks to the survey teams who dedicated their time and efforts to helping to collect data over the last 3 years. And I'm going to pass this over now. Let's see what time we're on. See if we can take some questions. Are there any questions? Just checking. Paula, did you want to ask a question?
No, I didn't.
Oh, you have your hand up. Okay, I'll put it down.
Sorry.
That's fine, don't worry. Mats Forsberg. Hello. Okay, so I'm going to take a picture. I can't hear anything there, so I'm going to take a question from Terry Lay here. Has the A68 iceberg affected the upwelling and food for whales? I think that's a really good question. It's quite early stage, and actually we have the South Georgia Science Manager here tonight, Martin Collins, who can probably comment better on the effect for other krill predators. But my understanding is that it's possible because there have been very few whales sighted this season. I think Martin Collins is going to talk about that a bit more in his talk tomorrow evening as well. But early news from this season is that there were very few whales. Whether that's due to the A68, it's a bit unclear at the moment because I think it's a bit too early to say, but it's quite possible, yes, because that iceberg was very large and it may have blocked some krill transport onto the island, onto the shelf waters. John, we're going to take we might take your question when we talk about blue whales later in the session. I hope that's okay. Kathy, the best time of year to see whales around South Georgia? That's a good question. It probably varies. We are mostly seeing them around January. February can be a bit more variable, and there's also a lot of whales caught historically in March time, so that may also be a good time to see them now, but our surveys are more in the summertime, so January would be a good bet. I'll take one more question from Mats Forsberg. Don't you get sightings from expedition ships? Well, actually, we have been very fortunate that expedition ships submit sightings to the South Georgia Museum, then we can compile them together. So really encourage any sightings to be reported to the South Georgia Museum because that's a really helpful way to get them into these summaries we're making of the whales sighted over time. Photo IDs gathered from expedition ships go into Happy Whale, and hopefully the photographs, because they provide an open access database for photos, which Ted Cheeseman's going to talk about in a bit more detail during our webinars later. Okay, we're out of time now for my questions, but I'm going to pass on to our next speaker tonight, and that's Emma Carroll. So Emma, can I ask you to screen share? Thank you.
Thanks, Jen. Can you guys hear me okay?
Yep.
Great. Right, so well, it's my morning here. So this morning I'm going to talk about the right whale genetic diversity and population structure across the South Atlantic and how South Georgia fits in with that. So, I'm hoping. Okay, so as Jen alluded to, southern right whales, like many other baleen whales, their populations are actually more of a series of migratory networks. So southern right whales have preferred winter breeding grounds where mums will have their babies and whales will socialise and breed. And then they'll migrate to their favourite feeding grounds and then back again. And so again, I'm from New Zealand, so a lot of my graphics are from my part of the world. And you know, we get these migratory movements, but it's a bit more complex than this kind of one-to-one that first graphic here shows. And that, you know, one wintering ground can have multiple feeding ground associations. Sometimes whales do move between different wintering grounds. And so you end up with this network of different wintering grounds and different feeding grounds with different levels of connectivity and movement between them. And so what genetics allows us to do is to understand where South Georgia sits in this migratory network and effectively understand which wintering grounds it's associated with, and also look at the level of genetic diversity in the South Georgia feeding grounds. And genetic diversity is important because high levels of genetic diversity are correlated with better population outcomes, and essentially populations with higher genetic diversity will persist for longer and be more resilient to environmental change and other challenges. So what's really unique about South Georgia is we were actually able to directly study a southern right whale feeding ground. This is the first time, well, one of the only times that this is really possible with southern right whales and a lot of other large baleen whales in the southern hemisphere, because you know, South Georgia is an island that we can be based at or be based around, as opposed to the difficulties in working around Antarctica. And we knew something about the connectivity of South Georgia to wintering grounds at the start of this program. So there have been links to the Argentinian wintering grounds using photo ID and satellite telemetry work. And previous genetic analysis suggested that there were links to Argentina and Brazil as well. And so the way that we went about doing this, I saw that we had a question about how we biopsy sample whales. Well, essentially, we use a modified crossbow or another system for deploying a biopsy tip. So a biopsy dart, this is a dart here. So the tip of the dart hits the whale and pulls out a plug of skin about the same size as your fingernail. And this can be quite a challenging enterprise to do, and especially on the high seas of South Georgia. But what that gives us is a small bit of tissue, skin tissue, which is what this picture shows here. And this tissue gives us really high quality DNA. And we call the genetic information that we get from this DNA a profile, because it's actually a composite of different kinds of genetic markers. And the first one I'm going to talk about is a maternal lineage. And so this is a mitochondrial DNA control region. And essentially, that is a type of DNA that is inherited from your mother. And what we do is we extract the DNA and then we sequence the individual base pairs of a particular region. And that gives us what we call a haplotype, or a different version. And then we can look at the frequencies of these different haplotypes in those wintering grounds and in South Georgia, and start to get an idea about how different those haplotype frequencies are. And the other type of DNA that I'm going to talk about today are biparentally inherited DNA markers from your nuclear genome. And so that's really useful because it tells you not just about those maternal patterns, but also the paternal patterns as well. And so we also characterize these markers, and then we look at the different frequencies, which is what is shown in these figures. And using these different frequencies of the different markers, we can generate what are called F statistics. And that tells us the degree of difference or differentiation between the different populations that we're studying. And that can go from 0, which is essentially no difference, to 0.05 or 0.1, which is moderate. And so it's a continuum, which allows us to compare these different populations. And so as part of this work, we were able to expand the comparison. So we've worked with project partners across the South Atlantic, and compared with previous studies, we were able to combine published data with new data from Brazil and South Africa, and more information from South Georgia. So we were able to use the 8 samples collected by Michael Moore and colleagues in 1997 and the 3 samples collected in 2018. And we were able to increase the number of markers compared with previous studies. So we were able to increase the number of nuclear DNA markers or microsatellite loci to 16. And we were also able to look at the mitochondrial control region, about 300 base pairs of it. And just a quick note about mitochondrial DNA. So as I said, it represents maternal lineages, and given and in my first slide, I talked about how females will have their calf on their preferred wintering ground, and that calf will travel with her to her favorite feeding ground and then often back again. In that way, the mitochondrial DNA represents a marker for the kind of inheritance or learned behaviors of these migratory destinations. And so as direct maternal descendants will share a mitochondrial haplotype, and maternal fidelity and site fidelity will influence the distribution of these haplotypes, we look at significant differences in haplotype frequencies across these migratory networks. We get insights into the behavior of these animals and how separated or connected they are. And previous work shown that there is significant differentiation amongst the wintering grounds. And so if we look at the mitochondrial F statistic, there's moderate differentiation in that mitochondrial DNA or maternal lineage markers around the world. And there is, weak difference based on those microsatellites or those biparentally inherited DNA markers. And when we add in the South Georgia data, and so first of all, we're going to talk about just which maternal lineages are found in South Georgia. And so this is a haplotypes network. And so each circle is a different haplotype or maternal lineage. The size of the circle represents the frequency of that particular lineage, and the color represents where that lineage is found. And what you can see is the Indo-Pacific is shown in yellow, and there's only about a handful of these different lineages in the Indo-Pacific, but some of them are really common. On the other hand, if you look at South Africa in blue, there are lots of different lineages found in South Africa, but they're often at lower frequency. And so that gives us insight into diversity. South Africa and Argentina have a lot more diversity of lineages than Australia and New Zealand. And we can also start looking at where South Georgia sits in this network. And what we can see is that, South Georgia has 9 different lineages, 5 of which are only found in the Southwest Atlantic, so Argentina and Brazil, based on the samples we have from those regions. 1 lineage is only shared so far with South Africa, and 3 are found in both South Africa and the Southwest Atlantic. And when we move from that network to these differentiation statistics, what we find is South Georgia is not significantly genetically different from Argentina and Brazil, so that Southwest Atlantic group, but it does show significant differentiation in those maternal lineages from South Africa. So that suggests that those kind of migratory patterns or migratory fidelity, those links are stronger with the Southwest Atlantic than they are with South Africa. When we look at the nuclear or biparentally inherited markers, we find a bit more of a complex pattern. So there's a lot going on here, but what this essentially shows is the probability of each DNA profile along this x-axis. It's the probability of each DNA profile marked by a dot. So the circles are DNA profiles of whales sampled in South Africa. The squares are DNA profiles of whales sampled in Brazil and Argentina. And the triangles are South Georgia. And essentially we're talking about how good a fit these DNA profiles are for South Africa and for the Southwest Atlantic. And what this line here is basically if the DNA profiles fit on this line, then they're equally good fit for South Africa and the Southwest Atlantic. And there's actually only a very small genetic difference between these two groups, the South Africa and the Southwest Atlantic. And if you take one individual and try and assign it back to sampling wintering ground of origin, you're only going to be able to do that correctly about 80% of the time. And so that's not that great. So we say there's only weak differentiation and moderate assignment probabilities. But interestingly, 9 out of 11 of the South Georgia samples are more likely to be from the South Atlantic than from Southwest Atlantic than from South Africa. And when we consider the data set as a whole, there's actually quite a high probability that they are all from the Southwest Atlantic. So despite this kind of low resolution power of the biparentally inherited genetic markers, we're actually able to say with reasonable confidence that genetically the samples from South Georgia are more closely associated with the Southwest Atlantic. And so just as a quick summary, we published this work in the Journal of Heredity in 2020. And as a group, we can say that South Georgia is more similar to the Southwest Atlantic. But as individuals, there's still a chance that some of those animals are coming from South Africa. And future work, we're also increasing our resolution power by increasing the number of markers from about 16 or 17 in this study to thousands from a different approach called RAD sequencing. And we've also genotyped the 9 unique whales from 2020. And we can say that there's 6 females and 3 males in that data set from 2020. And the other thing I wanted to highlight is that one of the samples collected in 2018 actually allowed us to sequence the whole genome of the southern right whale. So that was a big collaborative project involving project partners from this project, and also with DNA Zoo, which is an excellent organization and that is working with researchers around the world to sequence the genomes of many different species. And so that's a really exciting development that's kind of sprung out of this work, and that will enable us to understand things like how the evolutionary history of right whales has shaped things like their immune system, and how whaling might have impacted genetic diversity around the world as well. Oh, and just have to, sorry, I stopped before my acknowledgements, but yeah, I'd just like to acknowledge that this is a massive project that has involved dozens of people all around the world to make this happen.
Thanks very much, Emma. That's a really interesting summary. We've got time for a couple of questions. There's one from Jimmy commenting that he was in Wellington and saw a southern right whale in the harbour in Wellington, and wondered how far these animals migrate, and is it at all possible that you could get a Wellington animal in South Georgia?
Right, so I actually have created the DNA profile for that whale, the Matariki whale, because he was in New Zealand just as I moved back. So how far they go is a really good question. And so we've done some satellite tagging in South Georgia and also in New Zealand. And we know that the furthest we've got from our New Zealand whales is about 5,000 kilometres. So the whales swam from the Auckland Islands, which is 400 km south of New Zealand, halfway to South Africa and turned around. We probably don't have a big enough data set to really say if that's unusual or not. And whether things have changed since the whaling era, but we think it's unlikely that the whales from New Zealand and Australia are going into the South Atlantic. I didn't show it, but the genetic analysis shows there's actually a pretty good level of genetic difference between Indo-Pacific and South Atlantic.
Thanks very much, Emma. I think we're probably out of time for talked questions, but thanks for the Q&A. I'm seeing quite a few great questions coming in there. So we'll move on to Amy Kennedy next, who's going to talk about tracking whales at South Georgia, as well as the photo ID catalogue. And thanks very much, Emma, for your talk.
Okay, well, thanks, Emma and Jen. I'm just going to start right up here. I have a lot to cover. So let me share my screen. Okay, let's talk about whale tracking and photo ID results from South Georgia. Getting right into it, what everybody's been waiting for here are the South Georgia Catalogue results. Just very quickly, for those of you who don't know, we're rating vessel-based images. So unlike most of the catalogs that you've seen where they're aerial images of right whale heads, the images that we're looking at are taken from a vessel. And so each image that I receive is going to be...
I'm sorry, just to interrupt. You're not in presenter mode. I think you've got your tool, just so you know, just to go into the full slideshow.
Oh, I don't know how to do that. Okay.
You might need to, I don't know if you need to reshare, but
share screen. Okay. Sorry, good grief. And we practiced and everything.
Second time lucky.
Yep. Share screen. Let's try that. How's that?
Yep, we can see it.
Great. Sorry. Well, let's go here and talk about the South Georgia right whale catalog again. Back to these images here. So when I receive an image from a vessel, whether we took it or someone else, I give it a quality rating. Quality 0 through 3, 0 is the lowest and 3 is the highest. These ratings are based on oblique angle, focus, amount of visible velocity, and the exposure of the image. So 0, as you can see, none of those things align. Quality 3 is a very high or high quality, but anything basically a quality 2 or a 3 is considered high quality for us. Just to know that every image that I receive is given a catalog ID number, even if they're terrible images, but only right whales with a high quality left and a high quality right or an aerial image are considered definitely unique. So this is kind of confusing because we often can't match left-hand sides of the whales to the right-hand sides of the whales unless the photographer was very meticulous about taking those or taking notes on those images. And so in the catalog, individuals with only a left or a right may be represented twice in the catalog. And that probably happens quite a bit, but it's very difficult to say how much that happens without aerial images. So just to talk about the results quickly, I analyzed over 9,000 images from 2018 and 2020. Those were taken on our Bass right whale projects. In addition to that, there were nearly 400 additional opportunistic images submitted by naturalists and by folks on cruise ships, and also from bass researchers from other stations such as Bird Island and others. And those were submitted by a Happy Whale. These resulted in 76 assigned ID numbers. 29 of those had high quality lefts, 45 high quality rights, 7 high quality aerial photos. And that sort of distills itself down to 20 individuals in the catalog that are considered definitely unique. Again, there's probably more than that for sure, but it's very difficult to know how many the actual final number is unless we have a distinct match between the left head and the right head. But what happens here is that what we're ultimately trying to do is we had some success in matching. So finally this year I was able to make a match. As you can see in the red box, there was a whale photographed in the Gerlache Strait in 2011, just off the Antarctic Peninsula. And that same whale was photographed in 2019 off of South Georgia. There's two reasons why this is pretty exciting. One, because of the power of citizen science. Both of these images were submitted through Happy Whale by folks. The 2019 was submitted by naturalists from cruise ships. 2011 image was submitted by a passenger on a cruise ship vessel through Happy Whale. And I know Ted's going to talk a lot more about this, but this would not have been possible if people hadn't combed through their images of whales and submitted pictures to us. So I encourage you, anybody who's listening who may have been to these areas, to please look at your images, see if you have right whales with head images, send them to Happy Whale, and Ted will send them along to me to put through the catalog. The other thing that's remarkable about this is that southern right whales are rarely seen in the Antarctic Peninsula, and it was not known if those whales were part of the Chile-Peru population or whether they were part of the Southwest Atlantic population, of which South Georgia are part of. And so this photo ID connection supports the idea that the Antarctic Peninsula whales are coming, or at least some of them, are coming from the Atlantic rather than Pacific. And so that's kind of just the briefest summary of the photo ID catalog that we have going. And I'm going to move right into satellite telemetry results because I have a lot to talk about. We actually, in the course of this project, we tagged 4 whales. So we tagged 2 humpbacks and 2 right whales. And so I'm going to talk about the 2 humpback whales that we tagged in 2019. This is the overview of both of the full tracks, but I'm going to go into each one individually to tell you just sort of what each animal did as a descriptor. Let's talk about Alpha. So what I'm showing you here is just the localized movement that Alpha undertook. So from January 18th, which was the tagging day, to May 18th, that's 121 days. In that time, Alpha basically spent its entire time off of South Georgia waters. The maximum distance from tagging site was only 370 kilometers. And so for an animal that has no physical barriers to where it can go, it's choosing to stay in a remarkably small, incredibly small footprint, and really just sort of circling around the island for about 4 straight months. So that's more than half of the length of the tag was all spent off South Georgia. I applied a switching state space model to these data. And what that does is it categorizes each point as to whether it's considered travel movement or area restricted search movement. And area restricted search is more meandering, localized movement. And in the breeding ground, or in the feeding grounds, excuse me, it's synonymous with foraging events. And so if you look here where the red dots are, the red squares, you can see that there are at least 2 distinct areas of foraging or area restricted search that this animal undertook during its 4 months off of South Georgia. So now we're moving away from that localized movement, and I want to talk about the entire migratory and breeding ground movement. So I've color coded the tracks here: the northward migration, the north coastal, the south coastal, and the migration south. So let's get into it here. After spending 4 months off of South Georgia, on May 19th, the whale leaves the area. On June 21st, it arrives off the coast of Brazil. So during that time, that's a 34-day evolution, and it traveled about 4,000 kilometers, and that's at about 118 kilometers per day for that migration from South Georgia to the coast of Brazil, which is actually pretty fast for a humpback. Generally, they're somewhere around 90 to 100 kilometers per day, as you'll see. Let's look more closely at the breeding ground movement off the coastal shelf of Brazil. If we look at the orange track here, June 22nd to July 13th, the whale kind of just meanders along the shelf near Arroyos Bank. And July 13th, it reaches its northernmost point and then starts heading south again, almost fully retracing its steps. So the orange line and the purple line sort of kind of mirror each other on its way back south. August 3rd, about thereabouts, the whale starts to head offshore and start its southerly migration. So this coastal movement, the orange and purple lines, that's 43 days, roughly 2,700 kilometers, and 63 kilometers per day. And again, this is the distance I'm talking about are not straight line distances. This is point to point what the animal actually moved. So it's a little bit more meandering rather than just point A to point B. So about 63 kilometers per day during the breeding ground movement. And now we'll talk about the southern migration here, the green line. August 4th is when Alpha starts heading south. And then transmissions end, unfortunately, on September 3rd. But in that time, that's 31 days, the animal travels roughly 2,900 kilometers or 94 kilometers per day. And so just to put that into perspective, the data on the right are from Zerbini et al. He was able to tag a number of humpback whales off the coast of Brazil, of Abrolhos Bank. And the animals that he tracked, he calculated an average migration speed of 84 kilometers per day with a range of 63 to 100 kilometers per day. So if we look at alpha, the northern migration is 118 kilometers per day and southern is at 94. So alpha is traveling generally a little bit faster than the whales from Zerbini et al., but not anything that's too remarkable. So let's talk about beta. I know the lamest nicknames, but the other two get a little bit better, I think. Definitely every time I tag two whales in the same spot at the same time, they will always do the exact opposite thing, which is what we're seeing here. Tagged beta on January 22nd. Beta's tag only lasted for 54 days, but in that time, the whale traveled about 1,400 kilometers, ending up at Saunders Island on March 16th, where transmissions ended. So we're just looking at a zoomed in view of Zerbini et al.'s data. And you can see that definitely this whale in orange ended up basically in the same spot in Saunders Island. But the difference is that you can see Zerbini's data, the animals all bypass South Georgia entirely, and they approach the South Sandwich Islands from the eastern side of the island chain, whereas Beta went through the Scotia Sea and approached from the western side. So that's kind of the major difference between those guys there. Okay, moving right along here just to keep things going. Now I'm going to talk about the two right whales that we tagged in 2020, Annenkov and Braveheart. And just to take a little deeper dive here, we'll start with alphabetically. Annenkov, this whale transmitted for 117 days, so from January 28th to May 23rd. In that time, it traveled over 5,500 kilometers, but at an average about 47.7 kilometers per day. You can see the track is fairly meandering, doesn't seem to be in too much of a hurry. I would say that this is just exceptionally random. If you look really close at the month-by-month points, there doesn't seem to be a lot of movement correlation based on bottom features, or none that I can see that seems significant to me. Obviously, it means something to the whales, but just to point out here that the southernmost point that the whale reaches, March 31st, is at 66.2 degrees south, 41.6 degrees west before it starts heading north. So that's what we have for Annenkov. I also applied a switching state space model to these data. The points in red for Annenkov are areas restricted search regions or likely foraging regions. And again, I'm going to compare this to Zerbini beating us to it by tagging whales that also traveled to South Georgia in a different study. The scales are a little bit different on these two maps, but you can see that the areas that Zerbini et al. Highlighted as area restricted search here in the orange circles roughly correspond to areas where Annenkov was also engaging in area restricted search. And so while you don't really see much bathymetric ocean seafloor difference, there's clearly a lot of foraging potential in those areas. Okay, so now we're going to talk about Braveheart. This was our longest lasting tag. The tag transmitted for 238 days. But first, we'll talk about the highly localized movement of Braveheart here. So January 28th through July 9th, that's just over 5 months, 164 days. Braveheart spent its entire time just very, very close to South Georgia. The maximum distance from tagging site during this 5-month or 164-day period was 300 kilometers. Again, that's for an animal that can use the entire ocean. It's a really very small footprint that it's choosing to use. I think it's sort of worth pointing out that South Georgia receives an astonishing number of visitors. A lot of them are visiting ports to the north part of South Georgia Island. 72 visits in 2019, 10,000 plus passengers. People from literally all over the world are traveling down here to see whales, to see the sights of South Georgia. if a whale is not moving more than 300 kilometers in over 5 months, it's conceivable that it is there during the entire cruise ship season or the entire vessel season, which is something to think about. The whales really are just using a super small footprint, which is also tendency to overlap with vessel traffic, which I think Russell Leeper is going to talk about tomorrow. Now we're moving away from highly localized movement. We're going to talk about Braveheart's migratory movement. So after the 5.5 months at South Georgia, on July 10th, Braveheart starts to migrate. And from July 10th to August 5th, Braveheart travels from South Georgia to coastal Argentina and just travels north of the Falkland Islands and comes near the coast of Argentina on August 5th. So that's about 27 days, roughly 2,700 kilometers traveled, or about 100 kilometers per day for that northward migration. From August 6th to September 8th, Braveheart travels north across the coast. So if we're looking at the orange track here, Braveheart's northernmost destination is just north of Porto Alegre on September 8th. And that's 34 days of travel, around 2,300 kilometers for around 68.5 kilometers per day during its breeding ground coastal movement. And then finally, we got to track some of Braveheart's return trip. So after September 8th, Braveheart starts heading south, and transmissions end on September 20th. That's 13 days, 788 kilometers, or roughly 61 kilometers per day. But again, the coastal route towards the northeast and the return trip towards the south roughly mirror each other as the whale starts to head back. If we compare these tracks to those from Zerbini et al., first of all, it's important to know that he did all of his tagging in the Peninsula Valdes and Golfo San Mateus area, but the animals really heavily use that area in general, even outside of their tagging. So what is different about Braveheart here is that this whale just entirely bypassed the Peninsula Valdes and Gulf area and came close to shore at Rio Negro. And then the farthest northeast that the animal reached was Rio de la Plata. And you can see here that Braveheart bypasses that area altogether, travels the entire coast of Uruguay and up into Brazil. And so he's traveling a lot farther northeast along the coast than whales that have been previously tagged out of Argentina. And Braveheart is the only right whale that has been tracked visiting 3 countries in one breeding season. And so finally, just something, I realize that what I'm doing here is comparing one apple to one orange, but I do think it's remarkable that of the 2 animals that spent extended periods of time in South Georgia, if you plot their area restricted search, so we have Braveheart, the right whale, the pink diamonds, and Alpha, the humpback in red circles, the 2 areas of area restricted search of these 2 different species almost totally does not overlap. So they're really very segregated regions of area restricted search exhibited by a right whale versus a humpback. Again, it's one animal to one animal, but I think it's kind of remarkable. And I think it, warrants further investigation, whether they're just totally partitioning, whether it's normal that they do that, or whether there's just special about these 2 animals here. And so just really brief conclusions, humpbacks and right whales are showing very prolonged periods of a very localized residency time off of South Georgia. But in addition to that, they're also showing very long distance travel events. And so the 4 whales that were tagged in the study visited the exclusive economic zone of 6 different countries over their tagging time. And I think it's really important to understand that effective management needs to include a collaboration between the nations that these whales visit, but also between the nations that visit these whales. There's a lot of tourism interest in whale watching and ecotourism in South Georgia, and also throughout the breeding ground regions. And I think these things kind of need to mesh together for effective management of animals. And with that, in addition to Jen and Emma and their exceptional work and all the panelists that are talking today, I just have to say a special thanks to these folks. And Jen, I don't know if we have time for some questions, but I can try to answer some offline as well.
Thanks very much, Amy. That was absolutely fascinating. And I think, yes, we are a bit behind time. So in order to get everything in this evening, I'm going to move straight on to Susie. But definitely, I can see lots of questions coming in on the Q&A, which we can answer by text. And everybody, please, just to know, please, if you have questions for the panelists, please put them in the Q&A box, not in the chat, because we'll be able to pick them up in the Q&A, but we won't be picking them up in the chat so easily. Thanks very much. And I'm just going to move us on then next to Susie Calderan, who's going to talk to us about right whale acoustics. Thanks, Susie.
Now, is that my screen okay for everybody there?
That looks great.
Okay. So, good evening, everybody. Thanks very much for coming along. I would first like to thank all our funders and collaborators whose logos you'll see on the screen here. And I'm going to talk about southern right whale vocalisations on the foraging grounds in South Georgia. And so Russell Leeper and I have been the acoustics team detecting southern right whales off South Georgia, collecting the acoustic data and analysing the acoustic data from 2018 and 2020. And I'm going to talk to you about what we found out about southern right whale vocal behaviour at South Georgia. So the idea of using acoustics as part of the South Georgia right whale project to help find right whales came from work that Russell and I have been doing over the last nearly 10 years with Brian Miller at the Australian Antarctic Division as part of the Assoc project, where we've been using devices called dive-fast sonar buoys to locate and track Antarctic blue whales. So dive-fast sonar buoys are hydrophones, so they're underwater microphones that are self-contained, and when you throw them in the water, they transmit by VHF back to your vessel, and they transmit both the sounds that they're detecting, and they've got a magnetic compass in them, so they also tell you what direction those sounds are coming from. And they're actually military technology. They were developed by the military to find submarines. So the military throw them out of aircraft to detect submarines, but we've repurposed them to find whales. And we use these sonar buoys in conjunction with some software called PAMGuard, which we've specially configured. And we can use them to track whales over very, very long distances, specifically Antarctic blue whales. And so you can hear your whales from a long way away, and then you can follow the bearings that you're hearing the sounds on, and then you can find your whales. So they're a very efficient way of finding sparsely distributed whales over very large areas. Now, we decided that we'd use this technology at South Georgia, but we didn't know whether it would necessarily work so well with southern right whales as it would with Antarctic blue whales, because we didn't understand their vocal repertoire as well. We didn't know how much they would vocalise, how loud their vocalisations would be, and how well they'd propagate. So it was slightly experimental, but we wanted to maximise our chances of seeing as many right whales as we possibly could. And so we decided we'd use this dive-fast sonar buoy technology. So this is a dive-fast sonar buoy that I'm about to throw over the side right here. So they're quite small and they're quite self-contained. And as I said, you would launch that over the side, and then it would drop a hydrophone down and pop up and transmit back to your vessel what it hear and also the direction that the sounds are coming from. So you'll see that in 2018 we deployed 26 buoys, and in 2020 we deployed 31, and in 2018 there were 5 encounters where we had both visual and acoustics, and in 2020 we had 2. So sometimes we saw animals that we didn't hear. Sometimes we heard animals that we didn't see. But what was really useful was when we both heard and saw the same animal. And so the idea was to use these buoys to find the whales and then characterize their acoustic repertoire once we did find them. So here is a map showing where we deployed our sonar buoys and where we heard right whales. So you'll see that in 2018, we were deploying all our buoys around the north coast because that's where we were all the time. That's the red dots. And the open circles are deployed sonar buoys where we didn't hear any right whales. And the closed circles, the filled in dots are where we did hear right whales. So in 2018, we heard right whales on quite a lot of our sonar buoys. In 2020, the purple ones, we were deploying around the island because we were working around the island. And we didn't hear right whales on very many of our sonar buoys. And there's a few reasons for that, mostly because there weren't as many right whales in 2020 as there were in 2018. But also possibly because there were fewer animals, there was also less vocalization behavior overall, because a lot of their calls are contact calls. So that was where we deployed our buoys, and that was where we were hearing right whales in those 2 years. So what is it that we heard? Well, in the top left-hand corner here, you have a call that pretty much all right whales make everywhere. So all three, the North Atlantic right whales, Pacific right whales, and Southern right whales all make these calls, which is called an up call for obvious reasons. You can see on the spectrogram there of frequency against time that it's quite a low frequency call, which all right whale calls are. It goes from just below 100 hertz to just above 150 hertz. So it's low frequency, but it's audible. And they tend to make these very infrequently, certainly in South Georgia. And they just sound like little gulping noises. That's what we were hearing pretty much most of the time from our right whales. And then on the top right-hand corner, you'll see this broadband sound, which is a gunshot, which sounds exactly like you would think it sounds. It sounds like a gunshot. It's very loud and broadband, and they make them in association with mating. So those are the only two right whale sounds that we heard at South Georgia in both 2018 and 2020. Now, the bottom spectrogram is humpback whale call. So if you look at the plot again, frequency against time, you'll see that's much higher frequency. That's around 300, 350 hertz. It's much longer in duration than the right whale call. And also it's got harmonics, so you can see sort of replications of the call going up through the frequencies. So that's really useful that those calls are so distinctive from each other, because in unsupervised acoustic work, such as with long-term acoustic moorings, telling humpback whales and right whales apart can be problematic. So it was important that we were able to differentiate between them. And we found that we were able to do that in the encounters where we knew which way we could see the whales that we were hearing. So we were actually pretty pleased with how the acoustics element of this project went, in that we didn't know if we would be able to locate and track southern right whales as effectively as we were able to do with Antarctic blue whales. And we found that we were able to do that. There hadn't been any detection of southern right whales on high latitude feeding grounds with concurrent visual observations before. So that was a first in this project, and that was really good to be able to do. So we only detected upcalls and gunshots from right whales at South Georgia, which is quite a narrow repertoire, and we didn't have any circuits active groups there at the time, so it's not very surprising maybe that it was such a narrow repertoire, because often you get an association between lots of different behavioural states and different call types with right whales. And we also noted that the average frequency and duration of upcalls was broadly similar to southern right whales that are recorded in Argentina and the Auckland Islands, which is also not a massive surprise. Not dissimilar to North Atlantic right whales and North Pacific right whales, but a bit lower frequency than those. So the other thing that was interesting, as I noted in the previous slide, is that right whale and humpback whale calls at South Georgia can be clearly separated by frequency characteristics, by frequency sweep, by call duration, and by the presence of harmonics. So all the right humpback whale calls that we recorded were higher frequency than the right whale calls, and the right whale calls all had a frequency sweep, and they were much shorter than the humpback whale calls. So southern right whales at South Georgia are not particularly reliable vocalizers. We didn't detect lots and lots of calls from them. They weren't particularly easy to track, unlike Antarctic blue whales, which make very, very loud, reliable vocalizations and are much, much easier to track. But the fact that we could detect upcalls shows that there's a potential for long-term acoustic studies using moored recorders. And that's really useful for looking at studies of presence and absence on Antarctic and subantarctic breeding grounds and feeding grounds, and it will be really useful for interpreting data that we get from acoustic moorings which are not monitored. So it's been a really useful study looking at acoustics in South Georgia with the southern right whales, and we were also able to do acoustics with other whale species such as blue whales, which is something that I'll talk a little bit more about tomorrow.
Thanks very much, Susie. I think, given the time, I'll just take one quick question. Haley has asked, are you able to collect the buoys and reuse them?
No, dive-fast sonar buoys aren't reusable. And so that's something that we're working on. Well, we're working on reusable sonar buoys, and I think we've actually hopped through that technology, and probably we're going to move straight to trying to deploy and recover them from drones, which is probably going to be easier, because one of the good things about sonar buoys is you deploy them and then you don't have to go back and pick them up, so you don't use a whole load of ship time and fuel and etc. going and chasing your sonar buoys. So being able to deploy and recover them from a drone would keep the efficiency of the sonar buoy technology, but then you wouldn't end up with them in the water at the end.
Thanks very much, Susie. Okay, well, I can see there's some more questions in the Q&A, which Susie can follow up on afterwards.
Yeah,
so
do that.
Sorry?
I'll do that.
Thank you. We're going to move on next then to Dr. Gabriele Stowasser, who's going to talk to us about foraging ecology of right whales and other species.
Yeah, can you unshare, Susie?
Yeah, I thought I had. Hang on. Is that okay now?
Yes, thanks.
So hopefully, right. Come on. Oh, okay. I skipped the introductory slide. How do I go back in this?
I would just left click on your mouse.
Yeah, hang on, previous. Here we go. Right, so I work also at the British Antarctic Survey, and oh, hang on, I got the top bit missing here in my slides.
I can see trophic ecology of southern right and blue whales at the top.
Yeah, the problem is that I now have all the mute and everything stop video at the top of my screen, blocking half of it. Okay, never mind. Right. So I'm going to talk about the trophic ecology of the southern right whales and some of the blue whales in South Georgia waters. I'm a marine ecologist at BAS, and I work with stable isotopes and fatty acids to look at marine food webs and trophic ecology of marine species. So I'm going to give you a bit of the background, although Jen already touched on this, how my work comes into this. So South Georgia region was identified as a summer feeding ground for the southern right whales that are calving at the Peninsula Valdes in the winter. As you heard, they found that there was high calf mortality, but it couldn't be explained. So one of the suggestions was that environmental conditions at South Georgia would influence the whale population dynamics, in that whether they were successful in feeding there would be a factor influencing their reproductive rates. But before this project started, there was only opportunistic data on distribution, abundance of these whales, and actually no feeding ecology data in the Scotia Sea region. So stable isotopes is one of the biomarkers you can use to look at trophic ecology. And what we're mostly interested in trophic ecology are carbon and nitrogen. What we're interested in is, or what we're looking at, is a ratio of a heavy to the light isotope in nitrogen and carbon in the protein of the consumer tissue. As the prey gets eaten, more of the light isotopes is used in chemical reactions in the organism. And these light isotopes are then lost through respiration and excretion, which means that more of the heavy isotope stays behind and gets incorporated into the tissue of the consumer. And that in the end means that the isotope ratio of the consumer becomes heavier, more enriched, and compared to the prey. And that is about 3 to 4 per mil in nitrogen, and around 0 to 1 per mil in carbon. So we're using nitrogen to determine trophic level. If you look at a very simple food chain like you see here, you would have phytoplankton at the bottom of the food chain, which, for instance, have a value of 2 per mil. And then with each food trophic level, it increases. So the krill that feeds on the phytoplankton would have a nitrogen value of 5, and a whale feeding on that krill would have a nitrogen value of 8. Carbon, on the other hand, gives us information about the carbon source in the system at the base of the food web. So we could distinguish between freshwater and marine systems. We can distinguish if an animal's been feeding on the benthos or if it's been feeding in the pelagic, if it's been using more coastal areas or feeding more offshore. And what is really important for us in this study is that we can distinguish between latitudes, that is water masses. So a whale that's been feeding predominantly at the Antarctic Peninsula would have more depleted carbon values than, for instance, a whale that's been feeding predominantly around South Georgia. And again, if you had a whale that would be feeding predominantly on the South American shelf, that would be then more enriched than whales feeding in South Georgia or the Antarctic Peninsula. So this is the first data I will show you, which includes the potential prey species and the southern right whales biopsy samples from 2018 and one humpback whale sample from 2018. This is a typical graph of carbon plotted against nitrogen. As I said, so the nitrogen will give you a trophic position in the food web, whereas carbon will tell you something about the carbon source. So the closer the animals are grouped, the more likely is that they use the same carbon source. What we have here is, so you euphaosids in blue, you get the copepods in yellow, And you have some amphipods here. And what I indicated a bit as a darker blue is Euphosia superba, the most common krill species around South Georgia, the most numerous. And then I put in myctophids. They're not considered to be a potential prey species, but we know that these fish feed definitely on krill and copepods in this system. So I wanted to have them as an indicator species to see where the right whale would come out, and to just get an idea of the position. So as you can see, the southern right whales are slap bang. Here's my mouse. Are right slap bang within the myctophid, so where the krill and copepod feed. If you remember, nitrogen increases by about 3 to 4 between its prey and the tissue, then they could be well feeding on a mixture of krill and copepods here. And then we have the humpback whale here, which is on a similar trophic level than the right whale, but is interestingly more negative, more depleted in carbon than the right whales. And here I have to say, so what we have to remember here is that skin tissue in whales takes about weeks to months to assimilate the isotope values of their prey. So this suggests that this whale came from further south and still carries some of the isotope signatures it had from wherever it came. And if we then look at data that we collected, or that the guys collected in 2020, these are blue whale samples from 2020 in white, southern right whales in pink, and then we have the 2 southern right whales from 2018. So one thing is, you can see they fit, the southern right whales in 2018 fit very well with the animals from 2020. And apart from one southern right whale that has also more depleted carbon levels, the blue whales are quite separate, which again suggests to me that they came from latitudes further south, and they still carry a mixture of their isotopic signature from other regions. And the same thing might apply actually to these 2 outliers in the southern right whales, in that these high nitrogen levels, and particularly here the high carbon levels of these few, could well be that these are new arrivals from the South American shelf. Right, and then, so what we do have the prey data for 2020 as well, but this is still, it hasn't been measured yet. So this is still the data from 2018, and I superimposed all the whale species onto that. So basically, you have the 2 southern right whale species here, which are quite close together and fit very well with the prey species in the South Georgia region. Then you get the blue whales, which are more depleted, and the one humpback whale even more depleted, which suggests that they come from, or have the signature that is still from a region further south. And we do also have, I have to say, we have more humpback whale samples, which we will analyze in the future. And I think that was all the data. And I'd like to thank everybody who went out there and collected these samples, and everybody who helped finance this project. Thank you.
Thanks very much, Gabby. That was a really interesting talk, and it's great to see the blue whales in there. And I know that those stable isotope data are hot off the press from after a bit of a pandemic delay. So it's really nice to see this data growing for South Georgia right whales. Any questions? I think we had a question. John Barker asked, could we please define what trophic ecology is? Do you have a working definition?
Feeding ecology?
I guess it's about the food web, isn't it? Different level.
Yeah.
The different levels that animals sit in the food chain, which is what we're talking about.
Basically, yeah, if you're talking about the trophic level, it's basically what relationship these animals have to each other in a food web.
They get higher.
Thanks very much, Gabby. So I think we're going to move on now to Vicky, who's going to talk about
so you have to be able to
right wells at Peninsula.
No, well, you have to
share screen.
No.
Share screen.
Share screen there. Okay. There we go.
And it's which one?
It's right there.
Yep.
Share. There you go.
Hello there. Can you hear me? No.
Yep, can hear you great.
Okay, good. So we study the right whales at Peninsula Valdes, who that's off. It's off eastern Argentina. This is a mother calf pair. And I'm director of the program along with Mariano Cerrone, who's our director of research in our sister organization in Argentina. And Johnny, where did I advance it? Oh, this. Yeah.
No, wait. Why is it not?
Sorry, I got to advance the screen.
Why is it not? I do not know.
Okay, our study began in 1970 when Roger Payne was following up on people's comments that there were right whales off Argentina. And so he went down there and he studied the whales from the cliffs, and he realized that they had these white marks on their heads and that they were different in different whales, and that he could then follow the lives of known whales through their lifetimes. And so he started doing aerial surveys of the peninsula, and in 1971. So 2020 was our 50th year of doing surveys of the individuals there. But then along came COVID, and the government didn't give any permits for anybody to go out on the water. So we now have a break in our 50-year data set. So what I'm going to describe is the results from our latest survey and describe how our surveys work, and then analyses that were made by two members of our group on the photographs that were collected from boats off South Georgia. So the most recent thing we've done is finishing the aerial surveys of 2018. And when we collect the photographs, we fly along the perimeter of the two bays at Peninsula Valdes, and there's a note taker who and also the photographer, and they count the number of whales they're seeing. And so can I move this or not? Okay, this is the number of counts for each bay of mothers and calves from the aerial surveys, and then this is after, then we analyze all the photographs that we take and remove the picture, the best photograph of the whale, and we search for it in the catalog. And so this shows the ratio of the number of whales that are counted versus the number of whales that are identified in the population. So as of the end of 2018, the catalog now includes 2,921 known individuals. Roger's initial study is his thought was if we can follow individual whales throughout their lives, we'll learn much more about them than recovering tags like they used to do. And so now the next job is we're going to start doing the 2019 surveys. But now the part that relates to South Georgia is we have this problem at Peninsula Valdes that the whales are being attacked by kelp gulls. On the left is a picture of a calf, and it shows the circular wounds. Karina Groch, who's published in our group, who's published a paper on this, and you can see how they're circular. When the gulls attack the whales, they break through the top skin and make little things like the small circles, and then they aim from them, and they can keep on attacking whales for, I think we followed one for an hour that just kept it. They land in the water, and then when the next time the whale surfaces, they go down and attack them again. So this is the sort of thing, I think the question was, are there gull attack lesions on the whales off South Georgia? And so Flo Vilches and Karina Groch of our group down there, they analyzed 41 photographs that were good enough of the whales' heads, callosity patterns, to see whether there were any matches at Peninsula Valdes. And unfortunately, there weren't any matches found. But 10 of the photographs of the 77 they examined of the backs of the whales, and these are some examples. They blew it up, and so 10 of them had what is possible lesions. I myself, from having watched calves that have the marks like the calf on the left, when I see them come back as new as yearlings, it looks like you took a piece of sheet and put it over a glass. Looks like they in the next year look like a small lesion that is just flattened on the top, but doesn't have the kind of corrugation that is in these photographs. But this is all they found. So their conclusion was, well, no, they don't know what it is. I think we need to look at disease photographs that the whales can have, that may be pox marks or something. So that's all for me. Thank you.
Thanks very much, Vicky. That's really interesting, and it's a really impressive catalogue. Just great to understand that even though we don't have the linkages, that there were clearly whales using South Georgia waters that are coming from Peninsula Valdes.
Yes, absolutely. I mean, before, yes, and if you look at the stable isotopes in their skin, some of them feed and have in their baleen, you can see migratory patterns of them going to north and south in their annual migrations.
Thanks, Vicky. Okay, I'm going to see if we can move on now to Karina. She's having some trouble connecting, so Karina,
there she is.
Are you able to join, Karina?
Yes, I'm here. We are running out of energy, but I still have some battery left in my notebook and my cell phone, so I can do that.
Thank you. So Karina,
right.
talk to us about the Brazilian photo ID catalogue and matching with South Georgia.
Can you see my screen?
I can see the PowerPoint.
Okay, is that all right?
Perfect.
Okay, thank you, Jen, for this opportunity to talk about this very interesting result we had when comparing our catalogs, finally. So just a brief introduction about our study area. So we are in Brazil. We monitor through aerial surveys and land-based surveys the central southern coast of Santa Catarina state, which is in the very south of Brazil, where we found a main concentration area of right whales during the winter. So this is a calving ground where we have mostly modern calf pairs who come here to take care, to give birth and nurse their calves. We also have some solitary individual sightings and social groups, but this is not so common as modern calf pairs. So in recent years, we have been having mostly mothers, pregnant females that come here to have their babies. And the main concentration area, which you can see on the map, the light green line, is protected by a marine protected area that we proposed some 20 years ago to protect this species, which is still endangered here in the Brazilian coast. The species was under pressure of whaling for 400 years here in Brazil. So we have been doing the surveys through helicopters, and with the main purpose of taking photographs for their identifications. And since the surveys have been conducted since 1987, we missed some years along the year. In some years, we missed the surveys. We didn't have funding to do so. However, we have been doing mostly flights in September, which is the peak of the season. And we got by now some 1,024, more than 1,000 whales photo-identified in our catalog. We have many resightings, 3-year, 4-year Calvin's intervals, 5 years sometimes. We have some matches with Argentina. And we compared the catalogs then with the catalog from South Georgia. From the 76 whales that were individually identified or photographed of South Georgia, we were able to compare 17 aerial photographs because we only have photos from airplanes. We haven't been doing boat surveys here in recent years, although we have started to do again last year. So we were able to compare these photographs, the 17 photographs from which the 17 whales which had aerial photographs of right whales of South Georgia, and we found one match. It's the whale named with the number B139-02, which means that this whale was photographed and she was born in 2002, and she was sighted again. So she was photographed in the year of birth and resighted in 2005 and 2007. And in 2007, we saw her with her own calf. I understand that this is a 5 years of age. It's maybe too early for a whale to have a baby. I checked the data sheet many times to make sure that this calf was actually really from this whale. And this calf, I have a few pictures of the calf interacting with her. So I'm pretty confident that this baby was her baby. So this whale had
Karina, we might have lost the connection. I think we might have lost connection with Karina here, unfortunately. So that's a bit of a shame. I think in terms of
calf when she was 5 years.
Okay, I think we've lost Karina, unfortunately. If Karina can come back in due course, we'll have her back to finish off what she's talking about. But unfortunately, Karina, can you hear us?
I'm back. Yeah, just missed the connection. Just to finish. So this is a very interesting finding. And I would like to thank our sponsors, the Darwin Initiative and BEST, for supporting this comparison, this matching, this part of the South Georgia Right Whale project, and also Petrobras, the Brazilian oil company, which has been supporting our study, our monitoring in Brazil since last year. And is enabling us to continuing this long-term monitoring in Brazilian of right whales in Brazilian waters, which is very, very important for us. So that's it. Thank you.
Thanks very much, Karina. I think there's a quick question for you here from Peter Rex. Are there any good places where you can see the whales in certain areas or only directly out at sea in Brazil?
well, these whales here have a coastal distribution, so you can see them from the coast. I am here in my office in our Right Whale Center, which is located in the city of Imbituba in Santa Catarina State. It's just 100 kilometers south from Florianópolis, which is the capital of Santa Catarina State. And from the window of my office here, which is right here, I can see right whales during the winter. So you can see them here in the winter.
Thanks very much, Karina. So next we're going to go on to Els Vermeulen, who's going to present to us about matching with the South African catalogue.
Yes, good evening, Jen. All right, let me share my screen. Can you see the screen?
It says you've started screen sharing. I can't see a presentation yet. It's partly coming through. So maybe it's come through now, we can see it.
Okay. I can't see it myself.
No, I think it's just being slow to buffer, I guess.
Okay, yeah, maybe.
I'm sure it'll be up in a minute.
Let me just restart.
Yeah,
Sorry
maybe.
about that. Okay. See if this works. Can you see my screen?
I can see your screen. If this doesn't work, would you like me to put up slides for you?
Yeah, I think that could be best, Jen. You just put up the slides. It's only 2 slides, not a big problem.
Okay, give me a second.
Okay, that's better. So yeah, so in South Africa, aerial surveys for photo ID of southern right whales has started in 1979 by Professor Peter Best. So the aerial surveys itself started in 1969, and the photo ID component initiated 10 years later, since 1979. Just like in Brazil, these are helicopter surveys, which gives us the possibility to hover over the cow-calf pairs and photograph all the individuals with calves and all the animals that have a brindle coloration pattern. Up to date, we have 2,321 individuals in the catalogue, of which 1,839 reproducing females. So the survey runs on the southern Cape Coast, so basically between the area of Cape Town up to Nature's Valley, close to Plettenberg Bay, for those who know South Africa. Yes, Jen, you can go to the next slide. So we have also received photo ID images of 76 individuals from South Georgia, and we have selected 15 of these to pass through our system, which is a Hibby level system, which uses aerial photographs for photo IDing the right whales. So no match could be found with these 15 animals, but we do hope that with the future abilities from artificial intelligence for matching right whales, we can have more chances to ID more individuals. So I just gave some examples in these pictures below of the ones that I could use. To the left, it's relatively usable, but for me to pass it through the Hibble level system, I ideally can see both lip callosities or lips of the right whales. So obviously you can see the picture on the right, which I think was a drone image from South Georgia, that's a perfect image for us to pass through the system. So yeah, so far no matches found, but let's hope with some improved software in the future, yeah, we can do some more work on this. Thanks, Jen. That's all from my side.
Thanks very much, Els. That's a really nice summary. And yeah, I do hope we do find some matches one day, but perhaps we're not going to. With South Africa, who knows? So I'm going to move on next to Cecilia. And if we have any time at the end, I'll take any other additional questions about the photo ID catalogues. So Cecilia, I hope you are there. And
I'm here. Can you hear me?
yes, and Cecilia is going to talk to us about the Uruguayan right whale catalogue.
Okay, so can you see my screen or not yet? Not yet, I guess.
Not yet.
Okay. Now?
Yes.
Yes, okay. So I'm going to talk about the comparison of catalogs between Uruguay and South Georgia. This was done under the collaboration of 3 organizations or projects. That was Vida Silvestre Uruguay, Projeto Franca Austral, and Fauna Marina. So as you may know, Uruguay is located between the 30 and 35 degrees south, and that is in between 2 breeding areas, the Argentina Peninsula de Valdes and the one in Santa Catarina, Brazil. Studies done in Uruguay at the beginning of the 2000s showed that our coastal areas are not breeding, are mainly used for socializing and mating. So there's a few groups that can be seen of mother calf, but most of them are group socializing. Between 2001 and 2009, the Franca Austral project conducted aerial surveys following PAIN methodology. So we use an airplane that flew 220 kilometers of our Atlantic coast, not inside the Rio de la Plata. The flights were done at 500 meters high altitude, and they were done 5 to 6 times per season. Whenever we found a group of whales, of course, we fly at a lower height, between 150 and 200 meters, to take pictures of their heads. Most of the years where we used a film camera, it was ages ago. And then only since 2007, we started using digital cameras, and that improved the quality of our pictures. From that period, 2001 to 2009, we had classified 220 individuals. But what we did now for this work was we took all the pictures that were available, and we had some missings, and processed them using the one software called Discovery that allowed us to edit the pictures, filter them, and build a catalog. And then once we had that pictures in that catalog, we used the big fish to enter the images too. That allows you to enter all the callosity patterns. You specify one by one which you have there. So what we did, as everyone else said, we compare our catalog from that period to the catalog from South Georgia with the 76 pictures that they had. We also found the same problems that they have. Most of the pictures were from side view, and a few ones from above the head. All of the pictures we had were above the head, but were low quality compared with the ones from South Georgia. Unfortunately, this time we didn't find any matches. But we did found some what we call potential matches. That means that when we are filtering the data, that some individuals appear as potential matches. We checked we were 3 people trying to check if we could confirm or discard those individuals, but we couldn't. But we expect that in the future, if we continue with the comparison of catalogs, we would be able to find some matches. As we saw in the presentation of Amy with Braveheart, he passed by twice through Uruguayan waters last season, but we couldn't see it from shore. And so for the future, we have now pictures collected from Fauna Marina Uruguay since 2017. What they do is they conduct coastal surveys, looking from shore. If they find whales, they more or less do 100 days of search per year, and they more or less have like 50 days with sightseeing per season. So now we have all their pictures, and we are trying to incorporate it to our catalog. So we can integrate both time periods, and we hope in the future we will be able to find more matches with them. That's it. Thank you.
Thank you very much, Cecilia. I'm really glad that we're sharing photos, and hopefully we'll see some matches in due course.
Yes.
I think given the time right now, I'm going to move on to Jo Kershaw, who's going to talk to us next about Blubber and what we can learn from that. But I also just wanted to remind everybody there'll be 10 minutes after the end of our webinar, so we'll answer some questions again then as well. Okay, so have we got Jo there?
Yeah, I'm here, ready.
Thank you.
Cool. All right, share my screen. So can you see my screen, Jen?
Perfect.
Cool. Right, so good evening, everyone. I'm going to talk to you about some work that I did with the CMAM Research Unit at the University of St Andrews, where we were looking at blubber hormone concentrations in whale species that had been sampled around South Georgia. So I'll give you kind of a bit of an overview about some of the hormones that we're interested in and some of our preliminary results for our blubber biopsy results so far. So to start with then, in terms of this is what a blubber sample looks like. So what you're looking at here is a biopsy sample that was taken from a blue whale. So there in black and grey, you can see the skin of the animal, and there in pink and white, that's the blubber. So typically, as Emma mentioned earlier, blubber biopsies are taken using a modified crossbow and arrow system, where there's a small hollow stainless steel tip that's screwed into the end of the arrow. And then this penetrates through the skin and into the blubber and collects a sample before it bounces back off the animal and into the water, where it's retrieved, typically with a big net. So then once the sample's been retrieved, they were processed on board the research vessel. So this is the picture that was taken in January 2020. And here we can see Danielle and Daryl, so they're carefully processing the samples, where they'll measure them, and then they'll subsample them into different pieces that are then stored appropriately and then shipped off for analysis at various different labs for different kinds of analysis. So in St Andrews, we had the blubber part of the samples that were shipped to us for our hormone work. Then briefly then, in terms of the kinds of hormones that we're interested in, the first one is cortisol. So cortisol has got a number of different functions, but it's primarily involved in the regulation of fat metabolism or lipid metabolism. And generally, high cortisol levels are an indicator that the animal is metabolizing its energy reserves. So we can use it as a marker of overall physiological stress. So there's been previous work done on a number of different species that have found that there's a negative correlation between cortisol concentrations and morphometric indices of body condition and fat content. So generally, animals with high cortisol concentrations tend to be in poorer condition. So there was some recent work that's been done in humpback whales in Australia and also in North Atlantic right whales in the Northwest Atlantic that found that concentrations in the blubber of dead animals were significantly higher than concentrations in the live animals. And this is what we'd expect if we think that the dead animals are likely in poorer health, they're possibly diseased and in poor body condition. So for us, in our samples, in our biopsies, measuring the cortisol concentrations will give us an indicator of the overall physiological state of the individuals. And then our next hormone that we're interested in is progesterone. So in mammals, progesterone is responsible for maintaining pregnancy. So we can use this hormone to distinguish between pregnant and non-pregnant females, as concentrations are much higher in the pregnant females. So essentially, by taking these biopsy samples and quantifying progesterone, we can carry out a pregnancy test on the individuals. And so our previous work has shown that these concentrations of progesterone that are indicative of pregnancy tend to vary a little bit by species. So we've done some previous work in humpback whales, particularly in the Northwest Atlantic. So a study particularly looking at humpback whales in the Gulf of St. Lawrence in Canada, for example. Well, what we did was we used the blubber progesterone concentrations, and we were able to model the probability of the females being pregnant. So they were assigned a probability of 0 if they weren't pregnant or 1 if they were pregnant. And this method has been used in other study populations as well, in the Northwest Atlantic and the Gulf of Maine in America, and also a population of humpback whales along the Antarctic Peninsula as well. So it's really important to be able to identify pregnancy status in females, because we can then start to assess the pregnancy rates across the whole of the population. And by looking at pregnancy rates, we can look at reproductive success that can give us an idea of how healthy the population is overall, and potentially give us a feeling for population trajectories as well. And then finally, the last hormone that we're interested in is testosterone. So testosterone can be used to assess sexual maturity in males, and can also be used to assess the timing of reproductive cycles as well. So these methods have been used previously in fin whales in the Gulf of Mexico, and also in humpback whales in the Pacific, where they found that the testosterone profiles of males follow quite a predictable pattern for capital breeding species. So testosterone levels are fairly low, and then immediately prior to the breeding season, the testosterone concentrations start to increase, and then they remain fairly stable. So by looking at testosterone in our samples, we can improve our understanding of the timing of the reproductive events in our study species. And we can also start to look at things like potential migration timing and aggregations of sexually immature and immature males. So then in terms of the samples that we have so far, in 2020, there were 21 samples that were collected from 3 species, from blue whales, from humpback whales, and 1 sample that was collected from the southern right whale. And you can see, first of all, that the size of the samples varied quite a lot in terms of how big they were. So some of the largest samples were between 2 centimeters and 3 centimeters, for example, compared to some of the smaller ones that were less than ½ centimeter. So in terms of our work, in terms of the hormone work, essentially bigger samples are better, because it means that with smaller samples, we get more measurement error. So a lot of the work that we've been doing to date has been to try and assess what's the minimum amount of tissue that we need to make sure that our results are reliable, and therefore to make sure that we're maximizing the amount of information that we can get from these samples. So then, in terms of what we've been doing so far, then a lot of this work has been various quality assessment and quality control checks that we need to do when we're extracting hormones and quantifying hormones for the first time in new study species. So far, we've been able to validate our extraction methods. So that's how we're able to extract the hormones from the blubber tissue itself. And then we've also got ongoing work that's validating the quantification of these different hormones. And we do this using commercially available assays. So these are assays that are typically used to quantify these hormones in rodent species, for example, or in rabbits or in humans. So it's really important that we're able to validate them for these in whale samples to make sure we're able to quantify whale hormones, and that we're actually measuring what we think we're measuring. And then, as I mentioned, there's ongoing validation work in terms of what are the minimum sample masses of tissue that we need to make sure that our results are reliable. Then in terms of what we found then for the 3 different hormones and the 3 different species. So you can see in red are the humpback whales, in green is the southern right whale sample, and in blue are the blue whales. So for cortisol concentrations, these were very much within the range that had previously been measured in other species and in other populations. And we're not seeing any particular clustering so far in terms of some species being higher than others. For the progesterone concentrations, again, a similar story. These are very much within the ranges that have previously been recorded in different species. And what was interesting with the progesterone is that we had 1 female that had particularly high concentrations. So we've identified her as a pregnant female, and that was a blue whale. And then finally, in terms of the testosterone concentrations, so a lot of this still needs some extra validation and some still quality control and assessment checks. So we're going to try out some new assays to see if we can work these results out a little bit more and check that these particularly high concentrations, for example, in this blue whale and this humpback whale, to check that they're real and to check that they're not measurement artifact, essentially. So then in terms of what's next, the biggest thing now would be to combine these hormone data with the other data that have been collected on these individuals. So things like the trophic information data and the stable isotope data that we've heard about this evening. And together, these can give us a much better picture of and a complete picture of individual health. We'd also like to have a look at species-specific variation. So by combining these results with larger sample sizes, it'd be interesting to look at, are there differences between these 3 different species that can give us an indicator of their body condition, of population pregnancy rates, and the timing of their reproductive cycles that would be really important to understand how these animals are all using the same habitat and all feeding in the same area. And then finally, looking at population-specific variation. So comparing what we're seeing in the South Georgia animals compared to populations of the same species, but potentially with different population trajectories. So this is where I've cheated a little bit. So this is actually a picture of 2 North Atlantic right whales. These aren't southern right whales. And so North Atlantic right whales are an endangered species. So what would be really interesting would be to learn what we can about what we see in terms of hormone concentrations in the southern right whales that are species that are doing much better than the North Atlantic right whales, for example. And can this give us clues in terms of how potentially the North Atlantic right whales may be being impacted in different ways in terms of their overall health and their overall reproductive success? So thanks especially to all of the collaborators and to everybody who's gone out and collected these samples, and thanks to the funders as well. And I'm happy to answer any questions in the chat that you've got. Thanks.
Thanks very much, Jo. That's a great talk. I have one quick question I'll ask you, but then there are a few more great questions in the Q&A there. Antonia asks, how fast can hormones be detected in blubber after being produced?
That's a really good question, and it's still not completely understood what the kind of turnover times are. So in an ideal world, we'd be able to do studies with them and monitor them over long periods of time, the same animal, to look for how quickly we start to detect a signal. But obviously, we can't do this with big baleen whales. So there's been some studies in smaller cetaceans, so in odontocetes, that seem to suggest that anywhere between a couple of days and a couple of weeks, you're starting to see some of these signals. But in baleen whales, it's still kind of a question that's up for debate, really.
Thanks very much, Jo. That was really interesting. There's a few more questions for you to answer in the Q&A and then after the next talk. So I'm going to take the next talk from Morgana now, and she's going to talk about her work with Alex Aguilar and the University of Barcelona on using logbooks to reconstruct historic exploitation of right whales. Morgana, are you there?
Yes, I'm here.
Fantastic.
Can you see it?
Yes.
Is it in presentation mode? No, I think no.
It's not showing as the view yet.
Okay, now.
Oh, that's perfect.
Okay, cool. Thank you. So my name is Morgana Vighi, and I'm going to talk to you about a little bit about the reconstruction of right whale catches through whaling logbooks. This is a work that started many years ago during my doctorate, and I was able to produce some results thanks to the project, to the Darwin project, and thanks especially to Jen. So as it was mentioned before, right whales have been whaled during several centuries in the southwestern Atlantic. So for more or less 400 centuries now. But it was at the end of the 18th century that these activities started to have a very heavy impact on these species because of the introduction of the offshore open boat whaling. That was a technique of whaling that was based on the use of small rowing boats that were deployed from larger vessels that were sailing across the ocean during several months or several years. The main leader country of this activity was the United States, and also some whaling was done by the British and the French whalers in the area. But it was the American whaling that really impacted these species. So we can reconstruct the impact of whaling through different historical sources. But the main problem with these sources is that many of the data are just the overall discharges at port of the whaling products, so barrels of oil or baleen plates. So we don't have an exact allocation of these catches. We just know the overall number of products that were collected. Moreover, during whaling activity, especially in the 80th and 19th century, this activity was not so efficient. So a lot of individuals were struck, but not taken to the vessel. So they were lost at sea. Some of them survived, some others died. So in order to be able to refine a little bit this estimation, we went to the direct source of the data. So we went to consult the whaling logbooks. It is estimated that more or less whaling from American vessels were about 15,000 expeditions from the 1760s to the 1920s, and that more or less only 7,000 logbooks have survived to this date, either preserved in private or public repositories. So we decided to focus our study on the southwestern Atlantic Ocean, because we knew, according on historical sources, that there were 4 main whaling ground for right whales in this area. That were La Plata ground, Brazil banks, False banks, and Falkland Island ground. So we designed this study area, as you can see in the map, and we went to find the logbooks corresponding to voyages that were active in this area. What active means? Active means that these vessels were actively doing whaling in this area, so they were reporting either a sighting or a catch of some species. So this is a good example of a whaling logbook in which you can see the date, the position. You can almost read the letter. And so what we were extracting from the logbook was the daily the date, the daily position, if it was available. Sometimes they were not collecting the position every day, so we had to extrapolate, and all the whaling activities. So you can see in this example, you can read lower down 3 boats. That means that 3 boats were lowered from the main boat to go in pursuit of the whale. Also, you can see never got one. So these small vessels went to catch the whale, but didn't catch it. So we were taking note of all these activities, and to refine our analysis, we categorize a little bit this activity in the sightings, the lowerings, the catches that were the actual number of animals that were killed and taken to the boat for being processed. The whale struck and lost, so we don't know what happened to this whale, they were just lost. The killed whale after being struck, so these were struck but lost, dead. And some of these dead whales were recovered after a few days. So we also took note of this. We don't know if the recoveries were of the whale killed by the same boat or by another boat. This is something that we don't know. So we took note of for every year of activity that we had in the area of these numbers, and from them we calculated some whaling statistics as the loss rate factor. We calculate 3 possible factors. The most conservative one was assuming that all the struck whales were, that only the struck and killed whales were killed, actually. The less conservative one was assuming that all whales struck were dead. So we overestimate this factor, and then the average one assumes that only 50% of struck whales were killed. Then we also calculated the overall loss rate factor, considering all the years of activity in the area, and splitting it in two periods. So before the 1850s and after the 1850s, because we saw that there was very few sightings after this period, and we wanted to see what happened. So to provide you an overview of results, we collected information from 255 voyages over a period of over 150 years, more or less. To sum up, we had 20,000 active days in the area, approximately 2,500 right whale sightings, and then you can see the number of lowerings, the struck and lost whales, killed and lost, killed and processed, and the carcass recovered. Then we calculate the main values and range values for each year and overall across the study period. And to give you an idea visually of the distribution of the data, we have like the most of the catches distributed before the 1840, more or less. And you can see that for almost each successful catch, there were also losses every year of activity. So to investigate if there was a trend in these statistics, we use a Spearman's rank correlation test, and we found out that all the loss rate factors were actually decreasing across time, while the catch per lowered ratio, that was a ratio that we calculated to assess, to have an idea of the efficiency of whaling. So it was the number of caught whale every time that the boat was lowered. It was not decreasing, it was constant across the study period. We also investigate this trend with general additive models, including also other variables such as the effort, the type of the vessel, and the average size of vessels in terms of tonnage of the vessel. And again, we saw from the models that the only significant variables determining the variation in loss rate factors were the years, so the time. And so we had this the simplified model that was explaining almost 30% of the deviance. While for the catch per lowered ratio, we didn't have any significant variable, so there was no significant trend. So in conclusion, well, our data confirmed that this species was massively hunted in the southwestern ocean, and the concentration of these catches was in the first part of the 18th century. After the 19th century, sorry. After the 1850, there were no catches, but also there were no sightings, so no more whales in the area. Our loss rate factors were quite high in this first period. In some years, they were reaching a value of 3. It means that for each whale that were successfully caught, other 3 were lost, dead in the sea, or not dead but injured. So that also had an impact on this species. These values were decreasing over time, so we may assume that the efficiency of the technique was slightly improving. But our index that we consider to be reflective of the efficiency, that was the catch per lowering, was not increasing in the same way. We also have to consider that the number of catches in the second period consider was very low, so this statistic was maybe not significant for this reason. Finally, to provide to have an idea of the relevance of our results, because our logbooks were not randomly selected, we ran a random selection of voyages using the online database of whaling voyages. So we extracted 2,000 random voyages to check what proportion of voyages we had analyzed, and we found out that approximately 10% were considered in our analysis. So given the actual number of data that we had, we assume that our information is representative of the trends in whaling in that area. So this result provides this useful index that can be used to correct the estimates, the available estimates and future estimates of the whaling impact. And this is information that will be very useful for future assessment of the recovery of the population in this area and in other areas. So if you are interested in the study, there is a publication from last year, and you're welcome to contact me for any information. Thank you.
Thank you so much, Morgana. And I think we can't underestimate the amount of time and work it takes to go through all of those logbooks and try and make sense of broader patterns. It's a huge effort, and it's really nice to see the patterns showing up like that, just showing how inefficient whaling was at that time, and how much we need to take it into account, don't we, to in exploitation.
Yeah, sometimes they were only killing and losing and not taking home anything. So this had an impact, and it's useful to have an insight of that.
Yeah, thanks very much. So
thank you.
It's 9:07 here in the UK now, and we've just got a couple more minutes, and I wanted to take some of the questions that were in the Q&A for different panelists. And thank you to everybody who's been answering questions. I know the panelists have been answering questions behind the scenes. If you have any more questions, do put them in the Q&A box now, but I just will take a couple here. And there's a question to Vicky, if Vicky's still there, which is, can you tell the difference between gull scars and cookie cutter shark scars on the right whales at Peninsula Valdes?
Can you hear me?
Yes.
Oh, good. Yes. That's a great question. We know the gull scars enlarge with time, and it goes as deep as the blubber. And I think the sharks is more, I don't think you get something that enlarges slowly in a circle with time. I'm not sure, or whether people have done estimations like looking at the separation of teeth in orcas, and perhaps the cookie cutter sharks have a different size of a predictable size, not a wound that gets larger with time. I hope that helps.
Thanks. That's really helpful. We have a question from Mia, which is to Amy and myself, about whether there are krill data available from this season. Has this season been abundant for krill, and are there any humpback whales tagged this summer? So in terms of abundant krill, it's very early to have any conclusions about krill, but the recent surveys don't suggest that it is a good year for krill. So it's probably a poor krill year at South Georgia this season. And probably just for speed's sake, I can say there are no humpback whales tagged this summer. We don't have any research this season just gone, but hopefully we'll get down again in future, and we keep you updated about that. So another question, a question for Morgana here. Did hunting efficiency improve when they went from hand-thrown harpoons to gun harpoons to explosive head harpoons?
Yes, but that's not something that we can know from the right whale data, because as I mentioned, the right whale data were too scarce in the past, in the second half of the 19th century, to assume that. We did analyze also sperm whale data, and actually the efficiency was improved. So the same ratio, the catch per lowering ratio, was improving with time across the second part of the 19th century.
Thanks, Morgana. We have another question here about, it was quite a while ago, I'm afraid, but it was about the enforcement of fishing limits off South Georgia and just trying to understand how well the krill survey is monitored. There is a fishery patrol vessel that operates full-time at South Georgia in order to ensure that fishing is, illegal fishing doesn't take place there. I don't think I have any more to add to that, but if anyone on the panel wants to say anything. Martin?
Yeah, I mean, happy to add to that. I mean, the waters around South Georgia are kind of very well patrolled by the South Georgia government's patrol vessel Pharos. There are fisheries for krill, Patagonian toothfish, and mackerel ice fish around South Georgia. Actually, all those fisheries, at least in part, are certified by the Marine Stewardship Council as well. So they are considered, very well managed and sustainable. I know there was a question earlier about the level of krill fishing happening down in the Southern Ocean at the moment. All that krill fishing happens under the sort of management of the Commission for the Conservation of Antarctic Marine Living Resources, which is an international body, and they manage the krill fishery in the Southern Ocean. The fishery is, I think, reasonably well managed. I think there are concerns about the concentration of krill fishing in certain places, but generally speaking, there's certainly no illegal fishing or unregulated fishing for krill happening. It's all sort of fairly well recorded. There are observers on all the vessels now, so I think it's seen as being well managed. But of course, a key part of that management is that's taking that ecosystem approach to ensure that any krill fishing that does happen doesn't have an impact on the krill dependent predators, including the whales down there. So I hope that answers those two questions.
Thank you very much, Martin. That's a great answer. So with that, I'm going to wrap up our webinar for today and invite you all to join us for tomorrow night, when we're going to talk a little bit about right whales. We're going to include the right whale health story during the webinar, but we're also going to talk about other species, humpback whales and blue whales, and various discoveries we've made on those species at South Georgia tomorrow. So I really hope you can join us. And thanks very much for logging in. Do keep an eye on our updates on our Facebook group, which is SG Whale. And I will be posting a link to the recordings of these sessions onto that website as well, so that anybody who'd like to watch these later can catch up on them. Okay, well, thanks to you all. And thanks also to all the great panelists who've put together talks for this evening. It's been really nice to hear all the updates. Bye.
Bye. Bye, Jen. Bye, everybody.
Bye.
Bye. Thanks, Jen.
Pleasure. Bye.